This is DNA. You probably already know that it contains our genetic information. Therefore, you can say that it is like our blueprint that makes up you and me. You inherited it from your parents, which then they inherited it from your grandparents, which they inherited it from your great-grandparents, and then great-great-grandparents, and from great-great-great-grandparents. Well, you get the idea. This is what we call as transmission genetics, where an individual is the subject of interest. In population genetics, the subject of interest is not just one individual. Scientists are interested in the whole population, every member that is found in that population in fact. They study how genetic information is passed down from one generation to the next generation to the next generation and so on. What do they actually study? Genes. They study how genetic variation changes in a particular population over time, and if there are changes observed, what will be the cause behind it. In a simple word that you probably heard so many times before, evolution, as introduced by Darwin. Evolution is a change in frequency of a particular gene pool. Scientists use numbers or math a population or gene pool. What exactly is gene pool? Gene pool represents the whole genetic makeup for every member in a particular population. There are two types of frequencies to describe a population, the allele frequency and the genotype frequency. To explain genotype frequency, let me introduce you to a population of pea plants. In this population, there are pea plants that produce green seed color and there are pea plants that produce yellow seed color. If you can still recall that there are two types of allele, the dominant and the recessive one. In this population of pea plants, the yellow seed color is the dominant phenotype while the green seed color is the recessive phenotype. We can say that the color is determined by the alleles big Y and small y. The possible genotypes you can have in this population would be big Y, which indicates homozygous dominant, big Y, which indicates the heterozygous phenotype, and small y, which is the homozygous recessive. How can we calculate the genotype frequency? In that same population of pea plants, let's say that there are 8 homozygous dominant for the gene seed color, 64 heterozygous, and 128 homozygous recessive. Remember that there are 3 possible genotypes, big Y, small y, and small y. To calculate the frequency of each genotype, let me tell you what is genotype frequency. Genotype frequency is the number of individuals with one particular genotype. For example, it can be your eye color, blue or green, your hair color, black or red, hair types, curly or straight, plant seed color, anything. And you divide this number by the total number of individuals in the population. As for our example, the genotype that we are looking at is the seed color of the pea plants, yellow or green. Let's recall back the previous population of pea plant. There are 8 homozygous dominant, 64 heterozygotes, and 128 homozygous recessive. So how can we calculate the frequency of the homozygous dominant genotype? It's actually quite simple since we already have the information that we need. First, the total number of pea plants in the population is 8 plus 64 plus 128, which equals to 200 pea plants. Secondly, the number of pea plants that are homozygous dominant is 8. So to get the genotype frequency of big Y, you just have to divide 8 by 200, which will give you 0.04. Try to do the same to get the genotype frequency of heterozygous and homozygous recessive pea plants. You will have to divide the 64 heterozygous pea plants by 200, which will give you a frequency of 0.32. Finally, the genotype frequency of homozygous recessive will be 128 divided by 200, which equals to 0.64. What about allele frequency? Allele frequency is defined as the total number of one particular type of allele, either big Y or small y, and you divide it with the total number of all alleles in the gene pool. From our previous example, you know that there are 200 pea plants, but do you know how many alleles present in that population? Pea plants are diploid, like us. Since they have 2 alleles for the gene of their seed color, you just have to multiply 200 with 2 alleles, which will give you a total number of 400 alleles. Now, you need to get the total number of the dominant allele, which is the big Y, and the recessive allele, which is the small y, so that we can then calculate the allele frequency of both alleles. In order to calculate how many dominant allele, let's look at the information that we have. We know that there are 8 homozygous dominant pea plants and there are 64 heterozygous pea plants. So what you have to do is to multiply 8 with 2 allele, as each of these 8 pea plants has 2 dominant allele for the seed color gene. So now you have a total of 16 alleles. Now let's look at the 64 heterozygous pea plant. Unlike the homozygous dominant pea plants, the heterozygous have 1 dominant allele, the big Y, and 1 recessive allele, the small y. Therefore, you should always remember not to multiply the heterozygotes with 2. For genotype frequency of big Y, we are only concerned with the dominant allele. In other words, you can say that the 64 heterozygotes indicate that there are 64 dominant alleles and 64 recessive alleles. To get the frequency of dominant allele, just add 16 to 64 and you will get 80. Then divide 80 with 200 and you will get 0.2. Let's calculate the frequency of the recessive allele. Take the total number of pea plants with green seed color and multiply it with 2, which is 128 times 2 and you will get 256. Remember that you have another 64 recessive allele from the heterozygous pea plant. So you have to add 256 with 64, which equals to 320. Then divide 320 by 400 and you will get the allele frequency of small y, which is 0.8. The frequency of dominant allele is usually denoted by the letter P, while the frequency of recessive allele is denoted by Q. Frequency only range from 0 to 1. Therefore, if you know P, you can calculate Q. For instance, from the population of pea plant, the frequency of small y allele is 0.8. Therefore, you can calculate P by deducting 0.8 from 1, which will give you 0.2. What if the addition values of P and Q does not equal to 1? Well, that shows that there is something going on in that population. In the next module, you will learn about a mathematical model that is used to determine whether a population is at equilibrium or not.